Okay. Okay. Anybody else? She's telling me here there are two strands and they are helically coiled over each other. I call it as a single helix. Okay. When do I say it is a double helix? They consider the imaginary axis. Okay. Now the two strands of the DNA are coiled around each other, and both the strands are coiled around an imaginary axis. Okay. Like, I'm sorry, can I use your veiny? Can I use your veiny? Please, see. These are the two helix. These are the two helix. What is the two helix? I'm sorry, one helix. ये तीन छोटी का बनता है ना? ऐसे दो है. So I'm making a veiny out of how many? Two strands. Then I took my hand and I coiled her veiny around my hand. I hope you understood. So my hand is which axis? And now the two strands of the veiny are the two strands of the DNA. I coiled them. This is one helical turn. Then I took both the strands and coiled around my hand. So कितनी बार हेलाइसिस हुए? दो. एक बार जब दो पॉलीन्यूक्लियोटाइड स्ट्रैंड्स एक दूसरे के ऊपर कॉइल करते हैं, और दूसरी बार जब दोनों मिलके एक इमेजिनरी एक्सिस के ऊपर कॉइल करते हैं. That is what you mean by double helix. Understood what is double helix? Yes or no? How many polynucleotide strands are there? Two. Veiny has got how many strands? Three. Either it is because of a younger sister or somebody else that you know it. Okay? That a veiny has three strands, but my DNA has got two strands, which are coiled over each other. This is double helix or single helix? Now who is this? Now I took the two strands and coiled around the, and that is called as the structure of the double helix nature of the DNA. You understood my point, Muchilo? So do you see this red line? What do you saw it? What? Well, great vision, huh? Now who is this? Imaginary axis. Okay. And so you see the two strands coiling over each other. That is one helix. And now they are going on the either side of the imaginary axis. So that is what you call as the double helical nature of the DNA. Simple. Did you all follow what I said? Second property. The second property. It shows complementary base pair. It shows complementary. I like this word. Complementary base pairing. Very simple question. Yes, you got it. What is complementary base pairing? No? Sarah? Okay. That the purine always base pair with whom? Pyrimidines. And adenine base pair with two weak hydrogen bonds with thymine. Guanine base pairs with three weak hydrogen bonds with. This is called as complementary base pair. So the DNA follows a complementary base pairing ratio. इसमें ऐसा हुआ था कि रोजलिंड फ्रैंकलिन के स्टडीज के हिसाब से DNA had a constant diameter of 20 nanometers. वो जो दो स्ट्रैंड्स थे ना, उसके बीच में डायमीटर कैसा था? कांस्टेंट था. कितना चलो 20 नैनोमीटर्स. तो शुरुआत में जब वॉटसन एंड क्रिक डीएनए का मॉलिक्यूल बना रहे थे, तो कोई रीजन्स पे वो डीएनए बहुत ज्यादा ब्रॉड हो रहा था, कोई रीजन्स पे डीएनए बहुत ज्यादा नैरो हो रहा था। तो सोचो लोग क्या मिस्टेक करते होंगे अपना मॉडल बनाते वक्त, क्या मिस्टेक किया होगा लोगों ने शुरुआत में? किसके साथ बनाया होगा फिर? हाँ, प्यूरिन के साथ डाइसाइक्लिक है, बड़ा मॉलिक्यूल है, तो जहां जहां वो लोग प्यूरिन को बेस पेयर करते थे क्या होता था, वो डायमीटर बढ़ा देता था। जहां जहां पिरिमिडीन बेस पेयर करते डायमीटर क्या होता था, नैरो होता था। तो एज ए रिजल्ट ऑफ उसका कांस्टेंट डायमीटर नहीं था। तो वो लोग को क्या समझ में आया कि नहीं ये गलत है, हम लोग क्या करेंगे, प्यूरिन को किसके साथ बेस पेयर करेंगे, पिरिमिडीन को प्यूरिन के साथ। Then they started getting a constant diameter of 20 nanometer and they established the complementary base pairing. Okay, this is one of the unique discoveries in biology, because here you are trying to predict something which you have not seen, and you are turning out to be right at lot of fronts, which usually happens in other sciences, like maybe physics. Okay, where we work on a lot of prediction assumptions. Okay, and many a times they go wrong, but sometimes they come out to be correct. Got my point? So here also they did not see at that time, but they are predicting the model, and many of the points of the model are holding true. Okay, and one of them is what, Uchilo? Complementary base pairing rule. Purines pairing with pyrimidines, A with T and G with C. समझ में तुम लोग को? इस रूल को हम लोग बोलते हैं complementary base pairing rule. Then there is anti-parallel nature of the DNA. Which nature, Ucho? Anti-parallel nature. What is anti-parallel nature? Okay, one strand of DNA is read 3' to 5' ascending. If you remember, yesterday I made one 3' to 5'. You remember why I said 3', why I said 5'? Yes. And the other strand of the DNA is also 3' to 5', but that is going to be descending. So the two strands of DNA are parallel or anti-parallel to each other? Anti-parallel to each other. Got my point? And can I say here the orientation of oxygen is important, Bachelo? That decides whether it is an ascending strand or a descending strand. So one of the strand is 3' to 5' ascending, and the other strand is 3' to 5' descending. DNA is always to be read in which direction? 3' to 5'. Hello, sir. So the third rule is called as what? Anti-parallel nature of the DNA. Have you heard of this, Ucha? 4 gaps rule. सुना आप लोग ने? Yes, Tanvi, tell me. बोला? हाँ बोले इसके लिए. Just सुना आपने? Okay. हाँ, किसी ने सुना मुझे लोग? नहीं. It talks about purine-pyrimidine equality. It says that the ratio of purine is to pyrimidines is always 1. Okay, that means that A plus G upon C plus T will always be equal to 1. That is called which rule? Chargaff's rule. Make any model of a DNA, which look. I am writing a DNA molecule AT, AT, AAT, AT, AT, GC, GC, GC. Will the number of purine and pyrimidines always be the same? Yes. जितने प्यूरिन्स होंगे उतने ही क्या होंगे, pyrimidines. Agreed? Who are the purines? Adenine and guanine. Who are the pyrimidines? Thymine and cytosine. Count them, will always be the same. Got it? But A plus T upon G plus C need not be equal to 1. Did you understand my point over here? Like, look over here. Here A plus T upon G plus C, is it equal to 1? No. This is a DNA which is rich in 80 base pairs. There might be a DNA which is rich in GCP. A plus T upon G plus C more than 1 is an indication that it's a eukaryotic DNA. A plus T upon G plus C less than 1 is an indication of a prokaryotic DNA. You got it? I said A plus T upon G plus C is more than 1 is an indication of eukaryotic DNA. And less than 1 is an indication of a prokaryotic DNA. Okay? So will A plus G upon C plus T be equal to 1? Yes. Plus A plus T upon G plus C need not be equal to 1. It is constant for every species. Human beings, का A plus T upon G plus C ratio क्या है? Constant. आया बात समझ में? नक्की समझ में आया तुम लोग सबको? Then, next point. Can you tell me some properties of the DNA from your school or something you have heard before this? Tell me कोई property मालूम है आपको DNA का? हाँ? नहीं? Yes, Aditya? Base pairs? You have written this, huh? Complementary base pairs. Number of base pair per turn. Okay, okay. Chalo. We'll talk about now the physical properties of the DNA. Chalo. Okay. Now. Now you know the DNA has a constant diameter of how much, Muchilo? 20 nanometers. Okay. And let us observe here the two strands of DNA. Are they coiling over each other? No. The two strands of DNA are coiling over each other. And when they coil over each other, do you see they leave a minor chota khatta? And that is called as a minor groove. क्या दोस्तों? So when do you get a minor groove? When the two strands coil over each other. And when the strand coils over the imaginary axis, you get a bigger groove, and that is called as the major groove. So how many grooves do you get? 2. What are 2 grooves? Minor and major. When do you get a minor groove? When the 2 polynucleotide strands coil over each other, you get a, and when they coil over the imaginary axis, it's called the major groove. So who is this now? Major groove. And who is this? Minor groove. Once again, who is this? Major groove. Who is this, kids? Minor. Who is this? Minor. And who is this? Major. Major groove. Now did you understand what the minor groove and a major groove? Everybody? What's a minor groove and a major groove? When the 2 polynucleotide strands coil over each other, you get a minor groove. And when they coil around the imaginary axis, you get a major groove. Ayana. Now, the distance between 2 consecutive alternate major or minor groove is called, what is it called? Van Geyer or one spiral of DNA. क्या कहते हैं इसको? और one spiral of DNA. सुना ये वाला बात आप लोगों ने? So what do you mean one spiral या one gyre of DNA? The distance between two consecutive major या फिर minor. Got my point? And they show the presence of 10 base pairs. मतलब 20 nitrogenous bases. Okay? How many बचे लोग? 10 base pairs. मतलब nitrogenous bases. Okay. Now, so ध्यान में रखेंगे क्या ध्यान में रखेंगे? Minor groove, major groove, one spiral. Okay, and the angle between two base pairs, one above another, is about 36 degrees. So by the time you complete the 10 nitrogenous base pairs, you make a complete turn of 360 degrees. Understood? That is called as the pitch angle. क्या होता है उसको? Pitch angle of the DNA. So एक लैडर का एक रंग है, रंग ऑफ द लैडर मतलब एक नाइट्रोजनस बेस पेयर्स A और D, दूसरा कौन है? G और C. दोनों के बीच में एंगल कितना होगा? 36 डिग्री. तो जैसे जैसे हम ऊपर जाएंगे 10 के बाद एग्जैक्टली एक दूसरे के ऊपर आ जाएंगे. You will make a complete turn of how much? 360 degrees. So can I say it is representing a spiral staircase? It is representing a spiral staircase. Okay? तो कितने स्टेयरकेस के बाद ऊपर एग्जैक्टली वहीं पे आएंगे कुछ लोग? 10 के बाद. तो पूरा टर्न ले लेता है. परफेक्ट वन. Then the DNA shows the presence of one more property. It shows the presence of what would you? Palindromic sequences. What is a palindrome? कोई अच्छे स्कूल में गया? अच्छे इंग्लिश स्कूल में? पॉज करो. कोई अच्छे इंग्लिश स्कूल में गया? कल गया. मतलब here it is 20 Armstrong units, हाँ? नैनोमीटर में चाहिए. हाँ, palindrome में थे हम लोग. When we Julie said that it is something which can be read in the same way back and forth. Okay, so read this. Malayalam. Read it backwards. Such a sequence is called palindrome. Do you know of any other palindrome? Huh? Race car. How? Race car. Race car. हाँ, typical दिया देखा सकता है. Any more? Madam, Dad, Mom, Mandar. Read this sequence from 3' to 5'. G A A T T C. Read it from 3' to 5'.
G A A T T C.
So such a sequence is called palindromic sequence. Got my point? And when you will be taught biotechnology, this will be used. कहाँ यूज करेंगे हम लोग? अब ध्यान रखना, तब तक तो भूल नहीं जाना कि what are palindromic sequences in the DNA? The one which can be read in the same manner on both the strands on 3' to 5' directions. Okay, one of the example of a palindrome is GAATTC. GAATTC. Okay, क्योंकि कुछ एंजाइम्स होती हैं जो डीएनए को कट कर सकते हैं, and they exactly cut where they read this palindromes and they cut at certain regions. So this is important in biotech that the DNA shows the presence of palindromic sequences. Okay. Then regarding the physical properties, ये correction ध्यान में है तुम लोग के 20 nanometers नहीं, कितना है 20 angstroms है. Okay. I can add one more over here. How much is the distance between two nitrogenous bases? 3.4 angstroms. तो पूरे इसका लेंथ कितना होगा? 34 angstroms. Okay, and this you are talking about the B form of the DNA. Which form of the DNA? B form. How many forms of the DNA are there? A, B, C, D, Z, ऐसे अलग-अलग forms. Okay. The common one that we teach you is form. Which form? B form. Then the next property is something that Badudada read previously. Central dogma. क्या है?
Central dogma.
What do you mean by central dogma? That the DNA follows the central dogma. What do you mean by central dogma? Have you seen the movie Sholay, बच्चों?
Yes.
ये सब देखा, इतना पुराना पिक्चर 2 बार एनसीईआरटी पढ़ने को नहीं होती तेरे को, देखा? Right? What is the central dogma of the movie Sholay? कोई पढ़ के खाया नहीं? Right? I love to see that. Okay. बसंती बोल रहा है. The central dogma of Sholay is Thakur's revenge. Thakur wanted revenge against Gabbar. The whole movie is based upon that. But my point here, our entire movie of molecular biology is based upon this central dogma that the DNA can result in the formation of RNA, and the RNA can result in the formation of proteins. Okay, the DNA also has got autocatalytic functions, where the DNA can result in the formation of a molecule like itself, and that process is called as replication. Okay, the DNA results in the formation of an RNA by a process, which is called as transcription, and here it results in the formation of proteins by the process of translation. And these proteins are enzymes, and they can once again help in the formation of DNA and RNA. That is how the cellular machinery works. Okay. Now, we can get trapped in a question: what comes first, DNA or proteins? Which I'll not be able to answer. I hope you got my point. So, when the central dogma gets caught up in the theory of evolution, they say that for the first and the last time, there was formation of proteins without the DNA. Give me one miracle and explain the rest 100. You don't understand? Give me one miracle, and then I'll explain the rest 100. Okay? नहीं समझ होगा ना इंग्लिश? मतलब ये अपने को अज्यूम करके चलना पड़ेगा। क्या करना पड़ेगा? अज्यूम करके चलना पड़ेगा कि कुछ तो ऐसे इवेंट्स हुए, कुछ तो ऐसे इवेंट्स हुए। क्या हुए? लाहाजा, हाँ, मालूम कुछ तो हुए जिसके से पहली और आखिरी बार क्या हुआ बच्चों? प्रोटीन्स आ गए बिना डीएनए के। फिर उसके बाद ऐसा सेटिंग हो गया कि हमेशा क्या होगा? प्रोटीन बनाने के लिए क्या लगेगा? डीएनए। नहीं समझ में बात? इसको मैंने क्या बोला? मुझे एक मिरेकल दे दो मुझे, बाकी के 100 मिरेकल मैं साइंटिफिकली एक्सप्लेन करूं। ऐसे बोल रहा परीक्षा है क्या फायदा है? आप मेरा बात को समझ रहे हैं? NCERT is talking about the packaging of the DNA. See what figures are they giving there? For packaging, I want one of you to read it. Okay. See there. That who can read loudly? Yeah. Who will read? Yeah. Shall we read loudly a little bit?
Packaging of DNA helix. Taken the distance between two consecutive base pairs as 0.34 nanometers. If the length of DNA double helix in a typical mammalian cell is calculated,
0.34 nanometers. That would be 34 angstroms. Okay. Yeah.
simply by multiplying the total number of base pairs with distance between two consecutive base pairs, that is 6.6 into 10 raised to 9 base pairs into 3.4 into 10 raised to minus 9 meter per base pair, it comes out to be approximately 2.2 meter.
Okay. So what is the conclusion? That in a eukaryotic cell, the length of one DNA is so long. It is going to how much? 2.2 meters. I hope you understood the problem here. How much is the length? 2.2. आपका सेल तो 2 मीटर का है, कितना मीटर का है? 2.5 मीटर का है, हाँ? नहीं ना? So what is the problem? How do you explain that such a large molecule is going to get adjusted inside the cell का nucleus? Okay. That is what I am going to explain. See how to explain. For that, we want to talk about the packaging of the DNA molecule. Okay. What do you mean by packaging? It is going to be adjusted, packaged in such a manner that it can easily be kept, compressed, compacted inside the nucleus. That is called DNA packaging. Okay. So when a cell is not dividing, what did I say? When a cell is not dividing, what do you see? You see the presence of a nucleus. Yes or no? And I see the presence of beads on a string-like structure. What I see is the presence of which? Beads on a string-like structure. Right, kids? Beads on a string-like structure. Have you seen a rosary bead? Have you seen a Mangal Sutra? Alternative arrangement होना बहुत जरूरी रहता है ऑडियंस के हिसाब से, ठीक है? So आपने मंगलसूत्र देखा है सब लोग ने? उसमें स्ट्रिंग होता है और उसके अंदर क्या होती है? बीड्स होती कि नहीं होती है? हाँ, तो उसको हम क्या कहेंगे? Beads on a string-like structure. See figure 6.4b. Is it showing the presence of beads on a string-like structure? Yes or no? Ayana? These are the nucleosomes in the chromatin. What are the beads on a string-like structure? Nucleosomes in the chromatin. Do you see the entire structure? That is a chromatin thread. That is a or a chromatin fiber or chromatin, and it looks like the beads on the string like it represents the nucleosomes in the chromatin. Now you don't know what is nucleosomes, isn't it? आपको समझ में आ रहा है मैं क्या बता रहा हूँ? हमने पहले किसको देखा? सेल. सेल के अंदर क्या देखा हम लोग ने? न्यूक्लियस को. न्यूक्लियस के अंदर हम लोग ने क्या देखा? एक मंगलसूत्र जैसा छोटा सा पार्ट, बराबर, जो दिखता कैसे? बीड्स ऑन अ स्ट्रिंग लाइक. ओके. नाउ chemically what is it made up of? It is made up of DNA, RNA and proteins. What is it made up of? DNA, RNA, proteins. And proteins are of two types, guys. I think so you're aware of this. Non-histone and histones. Correct, Mr.? And the histone proteins are of five types: H2A, H2B, H3, H4, and H1. What are the five types of histone proteins?
H2A, H2B, H3, H4, H1.
Okay. And these histone proteins are rich in, tell me, they are rich in dash amino acids, basic amino acids, like dash and dash and arginine. Barabar? Ayana, what are they rich in? They are rich in basic amino acids like lysine and arginine. Underline that line in the text. Basic amino acids. Okay? And these all, they give a net positive charge to your histone proteins. Okay? So what is the chromatin made up of, Bachlo? It is made up of DNA, RNA and proteins. Proteins are of how many types? Two types. What are they? Histones and non-histones. Histones are H2A, H2B,
H3, H4, H1.
and they are rich in basic amino acids like arginine, and they get a net positive charge. They store the net positive charge. Shall I continue, guys? Are you all comfortable up to here? Okay, now see. Pay attention here. Huh? Two molecules of H2A. Can everybody see? Piche se bhi? How many molecules? Two molecules of H2A. Two molecules of H2B. Another two molecules of H3 and two more molecules of H4. So total how many molecules, guys? निकालो fingers निकालो देखो कितना है? 8 is called an octamer core. Now this core.
octamer core.
So what comes together to form the octamer core? H2A, H2B, H3, H4 come together to form an octamer core. Ayana? Okay. And on this octamer core, you have the DNA molecule wrapped around. A negatively charged DNA wrapped around tightly across a positively charged histone protein core. आया समझ में? And at the junction to keep it together is one more protein, which is called as the H1, and that results in the formation of one nucleosome. What is the result in the formation of?
One nucleosome.
One nucleosome. Ayana, such many nucleosome units, such many nucleosome units will be linked by the which DNA? Linker DNA. And on the histone core will be your core DNA. What's the packaging? Ayana. So you have the core DNA wrapped around the octamer, and between the two beads, I see the presence of which DNA? Linker DNA. And the whole thing will appear like a beads on a string structure. What are my points? Which is representing, what is it representing, Bachalo? It is representing the nucleosomes in the chromatid. What is my point? Now what is happening, guys, is who is this? Beads on a string-like structure, which is seen in a dividing cell or a non-dividing cell? In a non-dividing cell. आया ना? किसमें दिखता है अपने को ये? Non-dividing cell. When the cell is entering into the process of cell division, you guys know that धीरे धीरे the nuclear membrane will disappear, and we see the presence of structures which are called as chromosomes. Now what will happen, बच्चों? ये इतना लंबा molecule है ना, आया ना? It will start undergoing condensation, and this occurs by the loss of water in between. And धीरे धीरे it will undergo compaction. Okay, and finally it will change into what, Bachalo? It will change into what? Chromosome. So now the stages will be the DNA will form a nucleosome, which will form fibers called solenoids, which will form super solenoids, which will form chromatids, which will ultimately form
Chromosome.
and one chromosome will be able to carry how much? About 1400 nanometers of DNA. I hope you got my point. So individually DNA का कितना होगा? 2.2 meters. But जैसे जैसे उसको कॉम्पैक्ट करना स्टार्ट कर जाते हैं, बराबर है? 8 क्रोमोसोम के अंदर you can have a huge amount of DNA because of coiling and supercoiling. Have you seen a telephone wire? देखा है? पक्का? किसी मूवी में देखा है कि एक्चुअली में देखा उसने? हाँ, you है ना telephone wire? उसको अगर हम लोग ये करें मतलब extend करें, तो क्या होगा? लंबा हो जाएगा, बराबर है ना? लेकिन वो एकदम कॉम्पैक्ट हो जाता है बिकॉज ऑफ कॉइलिंग एंड सुपर कॉइलिंग. सो यू कैन इमेजिन द डीएनए मॉलिक्यूल inside your chromosome to be highly coiled and super coiled like a telephone wire. I hope you got my point. And that is called as the packaging of DNA. So how is the DNA packaged? By undergoing coiling and super coiling. It will form nucleosome, then it will form solenoids, super solenoids, chromatids, and the chromosomes. And how does a chromatin appear like? How does it, we see the presence of beads on string-like structure, and this is the nucleosomes in the chromatin. Okay, which is made up of DNA, RNA, and histone proteins and non-histone proteins. How many types of histones are there? H1, H2A, H2B, H3, H4. Two molecules each of H2B, H2A, H3 and H4 come together to form an octamer on which is wrapped the core DNA, which is tightly held in place with the help of H1. And you have a positively charged histone core and negatively charged DNA. So it is tightly wrapped. Lysine and arginine are the basic amino acids in the histone. Such many beads will be linked with the help of the linker DNA. That will be the nucleosomes in the chromatin. Are you following my point, Vichalo? Which will undergo condensation, coiling and super coiling to form nucleosomes, solenoids, super solenoids, chromatin and chromatid, and ultimately it will form a chromosome. Right, kids? Yes or no? And in 11th standard, you have already studied that chromatin is of how many types? 2 types. Yes. What are the 2 types of chromatin? Sister chromatin and non-sister chromatin. पढ़ाया गया ना तुम लोग को? कितने टाइप का क्रोमैटिन है? 2. कौन से 2 टाइप है? हाँ, सिस्टर chromatin or non-sister chromatin. बराबर है ना? हाँ? What is sister chromatin and non-sister chromatin? Yes, Anushka, what is sister chromatin and non-sister chromatin? Stand up, please. Huh? What is sister chromatin and non-sister chromatin? It is all bakwas. There is nothing like that. You idiots, what are you saying? Yes. There are only 2 types of chromatin. Sister and brother chromatin. No? अब पढ़ा मुट्ठी ला रहे बैठे बैठे. Yes, Anushka. Tan theta. Euchromatin and heterochromatin. Chromatin is of 2 types. What are the 2 types? Eu and hetero. Hetero is like you guys. It's junk DNA. Okay? Matlab it is genetically inactive. And euchromatin is genetically active. And majority of your DNA is heterochromatin. A small region is what? Euchromatin, as we will study in this chapter. आया समझ लो? ये पूरा क्या बताया मैंने, बताइए आज? बोलो. DNA, क्या DNA? Properties of the DNA and packaging of the DNA. So DNA, आपका नाम क्या है? राजेश. The DNA replication. The process of DNA replication will come under the autocatalytic function or the heterocatalytic function. Autocatalytic function. That the DNA can result in the formation of molecules like itself. Okay. Okay. So what are we talking about? DNA replication. What is DNA replication? When the DNA molecule results in the formation of a molecule like itself. The process is called as DNA replication. Okay. Is it on? Now, the method of DNA replication that we will discuss is called as semi-conservative method of DNA replication. Okay, and the idea was already proposed by Watson and Crick. Okay, see the sentence that they have given there. You got the Watson and Crick sentence statement. It has not escaped our notice. Yeah, shall we read that line loudly?
It has not escaped our notice that the specific query we have postulated immediately suggests a possible copying mechanism for the genetic material. Watson and Crick, 1960.
Yeah. Okay. That there is a possible copying mechanism of the DNA. Later on, after finishing this topic of DNA replication, we are going to talk about an experiment. Okay. Using a heavy isotope of nitrogen in a cesium chloride medium. जो बाद में आएगा. Okay. उससे हम लोग प्रूव करेंगे कि the DNA replication is semi-conservative. अगर सेमी कंजर्वेटिव का मीनिंग किसी ने मुझे पूछा तो मैं बोलता हूँ आधा बचाना. Half of it is conserved exactly. That is what you mean by semi-conservative. See, we all know the DNA molecule is double helix. Yes or no? At the time of DNA replication, the helix, it separates, or rather, I'll say it begins to separate from each other. Okay? And gradually, there is a synthesis of a new strand. And you'll see that the parental strands, the black ones, are the parental strands. They will separate from each other, and one strand will be newly synthesized. ऐसा समझ मैंने क्या बोला कि डीएनए रेप्लिकेशन के टाइम पे दो स्ट्रैंड्स हैं जो पैरेंटल स्ट्रैंड्स हैं वो एक दूसरे से क्या होने लगते हैं सेपरेट अनकॉइल होके और इमीडिएटली नया स्ट्रैंड क्या होता है सिंथेसाइज होता है एज अ रिजल्ट ऑफ व्हिच व्हेन यू गेट द फॉर्मेशन ऑफ अ daughter DNA, you will find that the daughter DNA has one strand exactly that of a parent and only one strand is newly synthesized. Got my point? So can I say half of the parental DNA is saved या फिर conserved in the daughter DNA? So what is the right word to be used? Semi-conservative method of DNA replication. आया समझ में? तो DNA के पास कितने strands होते हैं बच्चों? दो. DNA replication के टाइम पे दोनों strand एक दूसरे से क्या होते हैं? Separate होते हैं. और जो old strand है उसको टेम्पलेट जैसा यूज किया जाता है, ढांचे जैसा यूज किया जाता है और उसी के ऊपर एक नया स्ट्रैंड किया जाता है सिंथेसाइज. तो अभी जो नया डीएनए बनेगा उसमें भी दो स्ट्रैंड्स होंगे. एक क्या होगा पैरेंटल पुराना और एक ही क्या रहेगा नया सिंथेसाइज. तो पैरेंटल कितने स्ट्रैंड्स हुए? एक. तो आधा 50% एग्जैक्टली क्या हो गया सेम कंजर्व हो गया. That is what is called semi-conservative method of DNA replication. Do you get my point? That the old parental strand is going to be used as a क्या word use किया था हमने अभी template. You understood my point? It is going to be used as a template का हिंदी मीनिंग होता है ढांचा. एक ऐसा आकार जिसके ऊपर आप बाकी सारी चीज अभी क्या करने वाले हो बिल्ड करने वाले. So this model is called semi-conservative method of DNA replication. A eukaryotic DNA, bachcho is very long, as you have seen today. What my point? So what happens is the DNA has got many replication units, and one unit is called as a replisome. And we can understand this. Now imagine there is a cloth, एक कपड़ा, vertical कपड़ा. ऐसे में इमेजिनरी फिगर्स बोल रहा हूँ और वो कपड़े का लेंथ है समझो 1 किलोमीटर एंड आई वांट टू कट इट इनटू 2 हाफ्स. ओके, तो मैंने क्या किया, एक टेलर को बिठाया, एक टेलर को उसको बोला स्टार्ट टू एंड कट कर. व्हाट माय पॉइंट? तो पूरा चल चल करके एक ऐसा कतरी से वो कट कर. ये एक मेथड. या दूसरे मैंने आइडिया लगाया कि इतना लंबा डीएनए, सॉरी इतना लंबा क्लॉथ है, तो मैंने एक टेलर को रखा आफ्टर एवरी 100 meters. What my point? So एक टेलर किस पे? पॉइंट नंबर ए पे. वो कहाँ पे? स्टार्टिंग पॉइंट. नेक्स्ट टेलर कहाँ पे खड़ा है? बी. फिर ऐसे कितने टेलर लगेंगे मुझे अभी? 10 लगेंगे. आया ना? फिर मैंने विसल ब्लो किया. जैसे मैंने विसल सुना, सब लोगों ने कट करना क्या किया? स्टार्ट किया. तो टाइम कितना कम हो गया ना? So by the time the first tailor reaches from point A to B, the 9th one has already reached the 10th, and the 10th has done his job, and immediately you get 2. Alakshat? So what is easier? To cut the DNA, a eukaryotic DNA, from one end to another end as one unit, or you should have multiple units? Chalo. Alakshat? Aaya na? So that is why we say that a eukaryotic DNA has many origin of replication. It has got many origin of replication. In order to replicate the DNA, I need to first break this hydrogen bonds between the 2 polynucleotide strands. So then I'll nick them, I'll cut them, I'll give a nick. क्रिकेट में बोलो ना निक लगता है, पूरा बॉल लगता है कि हल्के से टच होके आता है बैट में से, और फिर विकेट कीपर के हाथ में तो हम लोग क्या बोल रहे हैं, निक लगती है, बोलते हैं कि नहीं, कट लगला. तो दैट इज हाउ हल्का कट दिया जाता है. किस बीच में? तो पॉलीन्यूक्लियोटाइड स्ट्रैंड्स के हाइड्रोजन बॉन्ड्स के बीच में. और उस निक को हम लोग बोलेंगे ओरिजिन ऑफ क्या बच्चे लोग? रेप्लिकेशन. तो ऐसे एक यूकैरियोटिक डीएनए में एक ओरिजिन ऑफ रेप्लिकेशन होंगे, मल्टीपल ओरिजिन ऑफ रेप्लिकेशन. हाँ. और एक यूनिट को हम लोग क्या बोलते हैं? रेप्लिसोम. दैट इज द कैरेक्टरिस्टिक फीचर ऑफ यूकैरियोटिक डीएनए रेप्लिकेशन. But my point that it has got multiple origin of replications and is going to be replicated in units, which is called as replisomes. तो एक डीएनए को मैंने कितने replisomes में भी डिवाइड किया? 3. तो by the time इसका एक का हो गया, ऊपर वाले का भी हो गया, और दोनों डीएनए सेपरेट होकर नया डीएनए फॉर्म किया. आया बात समझ में? Some of the steps that I can put in a stepwise manner for DNA replication would be: first, you need activation of nucleotides. Remember always, DNA replication is an energetically expensive process. It requires a lot of energy. And we can say the first step that is required is activate the nucleotides. You guys remember nucleotides, all of you? Yes or no? So we need to first do what? Activation of nucleotides. So we have deoxyadenosine monophosphate, deoxycytosine monophosphate, deoxyguanine monophosphate, deoxythymine monophosphate. Who are all these? Nucleotides. Where are they present? In the nucleoplasm. मैंने जानबूझ के पॉइंट मेंशन नहीं किया क्योंकि मुझे पता है आपको मालूम होगा, ऐसा मेरा अंदाजा है कि डीएनए रेप्लिकेशन occurs during dash phase of the cell cycle. Yes. Yes or no? Okay. So all this that I am teaching you is occurring during interphase. During which phase? Which phase of the interphase? S. You all know that. So we have these, what would you know? Nucleotides. They will be activated by the molecule. Who is the molecule, bachalo? ATP. Right, kids? In the presence of enzyme, which is called phosphorylase. क्या नाम है एंजाइम का? Phosphorylase. So it will result in the formation of deoxythymine triphosphate plus ATP will get converted to AMP. So I converted the nucleotides into a nucleotide triphosphate. What did I convert into, Chilo? Nucleotide triphosphate. I hope you understood. First it was a nucleotide monophosphate. ATP ने कितने phosphates दिया? 2. And I formed a triphosphate. अब ये खुद triphosphate molecule energy provide कर सकता है. आया बात समझ में? So अभी बाकी आपको पता है ये deoxyadenosine triphosphate plus AMP. ये क्या होगा? cytosine triphosphate plus AMP. ये क्या होगा? guanine triphosphate plus AM. I hope the class got my point, everybody. This is the first step. What is the first step? Activation of nucleotides. वो दो पहले सबसे पहले. आया ना? The second step is called origin of replication. What do you call the process?
Origin of replication.
Okay. So what is this origin of replication? That the DNA molecule is double helical. It is made up of 2 polynucleotide strands, and these 2 polynucleotide strands are interconnected by whom, bachalo? Hydrogen bonds. So there will be an enzyme whose name is helicase. क्या नाम उसका?
Helicase.
Yeah, helix destabilizing enzyme. कौन सी एंजाइम है?
Helix destabilizing enzyme.
ये हेलिक्स इसे स्टेबलाइज है ना, इसको क्या करना पड़ेगा अपने को? Destabilize. इसको कॉइल करना पड़ेगा कि अनकॉइलिंग करना पड़ेगा? Uncoil. So there will be a nick given. Nick मतलब cut. What is my point? It's a endonuclease activity. अंदर क्या किया उसको? Nick किया. Okay. And which enzyme did I mention, bachalo? Helicase. It will begin the unwinding of the DNA molecule. But what will happen now? The hydrogen bonds will be cut and the 2 strands will be uncoiled. Okay? As a result of which, the two strands, can I say, they will begin to separate from each other. They begin to separate from each other. Okay. Now sometimes what happens is, chalo, I am showing one strand now. Look here. As the strand is getting uncoiled, can I say it can form coils on itself, बच्चों? समझे मेरी बात को क्या बोला मैंने? वो स्ट्रैंड अनकॉइल हो रहा है ना? तो कभी-कभी वो स्ट्रैंड जब अनकॉइल हो रहे, कभी-कभी क्या हो जाता है बच्चों? वो खुद के ऊपर क्या ले लेता है? कॉइल्स ले लेता है. Okay? So I need to release this tension, which can be done by topoisomerase, or also called as what? Diaresis. They will give a cut, nick. As a result of what will happen, the strand will separate, and immediately it will cause it to join. You got my point, everybody? So these enzymes will help to relieve the tension during the process of what, bachalo? Unwinding. They help to relieve the tension at the time of unwinding. आया ध्यान में सबको? आया ना? तो सबसे पहले क्या हुआ? Origin of replication. जहाँ पे क्या किया गया? निक दिया गया. बराबर है? फिर बीच के हाइड्रोजन बॉन्ड्स को ब्रेक किया गया और अनवाइंडिंग किया गया with the help of helicase. Helix destabilizing protein. Okay? As a result of which the molecule is opening up now. Now there is a possibility that they can once again join. In order to prevent that, there will be another group of proteins which is called SSBPs. How is it called? SSBPs. Single strand binding proteins. How is it called?
Single strand binding proteins.
Single strand binding proteins. What will this single strand binding proteins do? They'll prevent the two strands from joining with each other. Okay? And what I'm seeing right now is, I see the presence of a DNA molecule which has opened like a fork, F-O-R-K, fork. Okay? And a fork which we use for eating. And this is called as the replication fork. क्या बोलते हैं उसको बच्चों? Replication fork. आया बात समझ में? Yes or no? Okay. Okay. So we have got the opening of the DNA strands, and is replicate. It is called which fork, bachlo? Replication fork. One thing need to understand, bachlo. As I am opening the two strands, those strands को क्या कर रहा हूँ मैं? Open कर रहा हूँ. Can I say this is the direction in which I am opening it? Yes or no?
Yes.
Yes. And if you want to read those two strands, this one will be read 3' to 5' towards the opening of the fork. Towards the
Opening of the fork.
yes or no? 3' to 5' towards the opening of the fork. But this strand will be read in the direction. In which direction, bachcho? 3' to 5' towards the opening or away from the opening of the
Away from the opening.
of the replication. Do you all understand up to the replication fork, everyone?
Yes.
Yes or no? Just read through the points for a couple of minutes. Recollect. काशी को अंकल सर मेरे होते हो आप दूसरों को अंकल कैसा बोलते हो? काशी को तुरंत बुरा लग जाता है काशी को। हाँ हाँ Gaji, what should be the next step? Whatever I wish. Okay. So the fourth step would be formation of a new strand. So the next step which look? Formation of a new strand. What is formation of a new strand? अब ये दो स्ट्रैंड एक दूसरे से क्या हो गए? सेपरेट हो गए। अब हमको क्या चाहिए? नया स्ट्रैंड का सिंथेसिस चाहिए। आया ना? The synthesis of the new strand requires a small RNA molecule which is called as a primer molecule. सुन रहे हो मेरी बात? Synthesis of a new strand will require a what does it require? Require a small RNA what bachalo molecule which is called as a RNA primer. Okay, and this RNA primer is synthesized by RNA polymerase enzyme. So कौन बना बच्चों?
RNA polymerase enzyme.
So what is an RNA primer? It is a small RNA molecule, oligonucleotide. Oligonucleotide. दीदी को एक बुक दे दो। मैं म्यूट कर रुक जाओ। मेरा सवाल है, ये क्या फॉर्म हुआ है हमारा? रेप्लिकेशन फॉर्म। अब हमको यहाँ पे नया स्ट्रैंड चाहिए डीएनए का। उसको बनाने के लिए हमको सबसे पहले क्या लगेगा? एक छोटा सा आरएनए मॉलिक्यूल जिसका नाम क्या बच्चों? आरएनए प्राइमर। उसको बनाता कौन है? RNA polymerase enzyme. Now this RNA primer will get attached to the 3' end of the separated template. Which end?
3'
Of the
separated.
So ये कौन है? ये ध्यान दो यहाँ पे, बोलो मुझे जरा फटाफट। ये कौन है? RNA primer, and it is synthesized in the 5' to 3' direction. खुद का कौन सा डायरेक्शन बच्चों? 5' to 3' डायरेक्शन। तो RNA प्राइमर टेम्पलेट के कौन से एंड पे अटैच होता है?
3'.
उसका खुद का सिंथेसिस कौन से डायरेक्शन में हुआ है?
5' to 3'.
वो कौन से एंड पे अटैच हुआ?
3'.
पूरा बोलो। 3' ऑफ टेम्पलेट। पूरा बोलो।
3' of template.
So where is the RNA primer getting attached?
3' of template.
So यहाँ पे सही जगह पे अटैच हो बच्चों। नेक्स्ट जो प्राइमर है दूसरा वाला वो यहाँ पे अटैच होना चाहिए। नहीं सर, गलत हो जाएगा। क्यों गलत हो जाएगा? कि ये टेम्पलेट का 5' है, उसका खुद का सिंथेसिस किस पे होना चाहिए? 5' to 3'. तो 5' तो नहीं हो सकता ना यहाँ पे, तो कहाँ से होगा? उधर। आपको समझ में आ रही मेरी बात? हाँ जोशी मैडम, लेट्स बढ़े आगे चलो। आया ना? पार्क को जरा उठाओ वहां पे। तो देखिए ये एक स्ट्रैंड है, ये क्या है? स्ट्रैंड है। देखो कितना न्यूक्लियोटाइड चाहिए आप लोग को? बस हो गया? थोड़ा रफ भी बना देना है मतलब ना, क्या समझ में आ जाएगा आपको? बस चलो लिखवा देते हैं। बीच में कौन आएगा बच्चों? फॉस्फो डाइस्टर क्या हो चलो बॉन्ड लिंकेज। ये कौन सा एंड है गाइस? 3 डैश। ये कौन सा एंड है? 5 डैश। ओके, डोंट स्टार्ट ओवर हियर बट दैट्स इट। ओके, कौन सा आपको नाइट्रोजन बेस पेयर चाहिए बताइए? हाँ जोशी आप बताइए कौन सा चाहिए आपको? चलो जी सी सी ए टी जी, ठीक है आया ना? हाँ। अब कौन सा मॉलिक्यूल आया गाइस? कौन सा मॉलिक्यूल आया? प्राइमर आया। आरएनए प्राइमर कहाँ पे? यहाँ ऊपर अटैच होगा 5 डैश पे? नहीं, कहाँ पे? 3 डैश। ये क्या होगा? सी। ये क्या होगा बच्चों? जी। बराबर कि नहीं? आया ना? इसमें और इसके बीच में क्या फर्क है? यहाँ पे सेकेंड कार्बन एटम पे आपको क्या मिलेगा? OH मिलेगा। मतलब ये कौन सा मॉलिक्यूल है? रेड वाला आरएनए है। ओके, क्लियर एवरीबॉडी? नेक्स्ट स्टेप in the synthesis is now there will come an enzyme which is called as DNA polymerase enzyme. Which enzyme?
DNA polymerase enzyme.
Which can cause polymerization of nucleotides. It can cause what?
Polymerization of nucleotides.
It can attract complementary nucleotides and bring about their polymerization. Imagine हम लोग सबको मिलके ना एक साखली बनाने का, चेन बनाना, तो गोल 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 बहुत सारे सर्कल्स, समझ मेरा बात? तो एक एंजाइम आएगी जो सारे सर्कल्स को साथ में लेके आएगी और उनको किस में ज्वाइन करेगी? एक चेन साखली में। आज समझ में? अब वो पूरी साखली का नाम क्या है? डीएनए। उसके एक यूनिट का नाम क्या है? न्यूक्लियोटाइड्स। और वो ज्वाइन करने वाले बॉन्ड का नाम क्या है? फॉस्फोडाइस्टर बॉन्ड्स। तो कॉम्प्लीमेंट्री न्यूक्लियोटाइड्स को अट्रैक्ट करना और फॉस्फोडाइस्टर लिंकेजेस को एस्टैब्लिश करना, ये कौन से एंजाइम का काम है बच्चों? डीएनए पॉलीमरेज। कौन सा?
DNA polymerase.
प्रोकैरियोट्स में 3 डीएनए पॉलीमरेज होता है। डीएनए पॉल 1, पॉल 2, पॉल 3। डीएनए पॉलीमरेज 1, डीएनए पॉलीमरेज 2, डीएनए पॉलीमरेज 3। इसमें सबसे मेन एंजाइम है डीएनए पॉलीमरेज 3। ओके, अब डीएनए पॉलीमरेज ने क्या किया? यहां पे कौन है बच्चों? सी। तो किसको अट्रैक्ट करके लेके आया वो? बोलो, जी को। आया ना? अब उसने क्या किया? It formed a phosphodiester linkage with the third carbon atom of your RNA primer. I hope you got my point. Then ऐसा कंटिन्यूस होते जाएगा? Yes or no, guys? अरे give me yes or no, boys यार. It's okay for the rough diagram? A and C. Now, my question to you guys is, the new strand is synthesized in which direction? The new strand is synthesized in which direction? 5 prime to 3 prime of its own. 5 prime to 3 prime of its own. बनने के बाद उसको आप कैसे रीड करोगे? 5 prime to 3 prime या 3 prime to 5 prime? लेकिन वो बन कैसे रहा है?
5 prime to 3 prime.
As always, in the next step now, another group of enzyme will come. It will remove the primer, because the primer is which molecule? RNA molecule. And then what will be synthesized? A DNA molecule. And then the process will be complete. आया बात समझ में? I repeat the last one again. So formation of a new strands require a small RNA molecule called as the primer. ये स्टेप के बाद कौन सा स्टेप आएगा? DNA polymerase attracts. What does it attracts? It attracts complementary nucleotides and forms what? Phosphodiester linkages. And forms phosphodiester linkages. PDL लिख दिया मैंने, आप लिख देना phosphodiester linkages. फिर क्या होगा ये बच्चों? And there is a synthesis of new strand in which direction? 5' to 3' of its own direction. Finally क्या होगा बच्चों? There will be removal of the primer और उसकी जगह पे क्या लगाया जाएगा? DNA molecule. And final step में कुछ enzyme होगी that will be doing proofreading. What will they be doing? After you write a paragraph, you proofread it for any mistakes. So if 1 in a million time अगर G के बदले C आ गया या फिर A है, अब मैंने देखो मेरा प्रूफ रीडिंग करना पड़ेगा. मैंने यहाँ पे A के सामने क्या लिखा? A है. ये सही है कि गलत है? तो एक बार वो एंजाइम्स क्या करेंगे? उसको पूरे हेलिक्स को रीड करेंगे. अगर A के सामने A है तो फिर से क्या डाल देंगे बच्चों? D. Got my point? So your DNA will be efficiently proof read. And any mistakes that have been incorporated at the time of replication will be rectified. I hope you got my point, everybody. Yes or no? Now there is one more concept over here, a good concept of leading strand and a lagging strand. Which strand bachlo? Leading strand and a lagging strand. See how I explain this point. देखो बच्चों, जरा ध्यान देंगे यहाँ पे. ये कौन है? रेप्लिकेशन फोर्क है. इसको कैसे रीड करेंगे? 3' to 5' towards the opening of the fork. और इसको कैसे रीड करेंगे? 3' to 5' away from the replication. Correct? Now, who is this? Primer. Who is this? Primer. The primer will bring about the synthesis of a new strand. In which direction? 5' to 3'. Is my arrow correct बच्चों? 5' to 3'. सही है, ध्यान देना यहाँ पे. अब जब रेप्लिकेशन फोर्क और खुलेगा कि नहीं और? तो नेक्स्ट डायग्राम में मैं थोड़ा सा ऐसे बड़ा बना. Got my point? ये वाला जो स्ट्रैंड है which is opened in the direction 3' to 5' towards the replication fork, उसका ये रेप्लिकेशन हो गया ऐसा आया ना. लेकिन जो दूसरा वाला है गाइस, उसने एक प्रॉब्लम एनकाउंटर किया. बताओ क्या प्रॉब्लम एनकाउंटर किया उसने? कि उसने भी सिंथेसिस किया 5 to 3 प्राइम में, but is it in the direction towards the opening or away from the opening? Away. तो देखो वहां पे गैप रह गया. I hope you understood my point. Yes. Now see what is happening. Who is this? Another guy. Who is this? Strand. Ayana? Yes or no?
Yes.
So can I say one of the strand is continuous, whereas the other strand is discontinuous? Got it? If I draw one more time, just to put the idea into your head, इतना ही कि और ओपन हो जाएगा बच्चों. और वन ओपन नहीं. ये कौन है बच्चों? प्राइमर. इसे कैसा सिंथेसिस किया? Towards the opening of the fork. So how was the synthesis? Continuous or discontinuous? Continuous. Who is this? One primer. It will produce a strand. Now there is another one. जो यहाँ था. Now what has to be done, बच्चों? Another. Correct? Ayana. And what is this now? समझ में आया concept? Yes or no? So which strand will finish the production of the daughter strand faster? Faster किसका होगा? Continuous या discontinuous? तो बार बार उसके फिर Are you high on something? ये continuous हो रहा है. ये कैसा है? Discontinuous. तो जल्दी कौन सा होगा? Discontinuous हो रहा है भाई. दूसरा दिला दिया. तो इसको क्या बोलेंगे? लीडिंग टेम्पलेट. क्या बोलेंगे इसको?
लीडिंग टेम्पलेट.
और ये वाले स्ट्रैंड को क्या बोलेंगे बच्चों? लीडिंग स्ट्रैंड. क्या बोलेंगे उसको?
लीडिंग स्ट्रैंड.
और वो दूसरे वाले को क्या कहेंगे हम लोग? क्या कहेंगे? लैगिंग टेम्पलेट. और ये स्ट्रैंड को क्या कहेंगे बच्चों? विल कॉल इट एज अ लैगिंग स्ट्रैंड. आया ना? और वो जो शॉर्ट सेगमेंट्स है, अब यहाँ पे कितने सेगमेंट्स बनाए मैंने? 3. These are called Okazaki fragments. क्या बोले उसको?
Okazaki fragments.
क्या बोले उसको?
Okazaki fragments.
समझा? Okazaki. क्या बोलेगा?
Okazaki.
अगर ये मुद्दा अगर उसको डिस्कवर कर दी तो उसका नाम क्या देते हैं वो? कोरे फ्रैगमेंट्स. अभी अंकल का नाम क्या था? ओकाज़ाकी. तो क्या नाम गया? ओकाज़ाकी फ्रैगमेंट्स. अभी बीच में प्राइमर्स है कि नहीं? रखना है कि रिमूव करना है? और फिर जो गैप्स आएंगे उसको भी तो फिल करना है. And those will be filled by enzyme called ligase. Ligase will rejoin the DNA. Got my point? So one strand is going to be synthesized in which manner? Continuous manner. And the other strand will be synthesized in which manner? Discontinuous. The discontinuous fragments are called which fragments? Okazaki fragments, or the lagging strands, which will be joined by ligases. तो पहले वाला कौन सा यूज हुई थी एंजाइम? फॉस्फोराइलेज, एंडोन्यूक्लिएज, हेलिकेज, फिर सिंगल स्ट्रैंड बाइंडिंग प्रोटीन्स, फिर आरएनए पॉलीमरेज फॉर प्राइमर, डीएनए पॉलीमरेज एंड लाइगेज, एंड आई हैव मिस्ड गायरेज और टोपोआइसोमरेज. ज्यादा नहीं But did you understand why do you get those lagging strands? Because one strand is synthesized in the direction towards the opening and the other is synthesized in the direction
Away from the
so that is why you get Okazaki fragments. आया समझ में? सबको समझ में आ मेरा point? Yes or no? हाँ? Just switch it off and we'll revise now everything.